I'm Casper Goverde, and I came here yesterday by train from the beautiful city of Lausanne. So this is an overview of our campus, and we are located in this building right here. And I am a member or a PhD student in the Laboratory for Protein Design and Immunoengineering, LPDI. Under the supervision of Professor Bruno Correa. And today I'm going to be talking to you about the design of soluble analog of integral membrane proteins, because this is what we do. We are protein designers. So let's first have a quick overview of what membrane proteins exactly are. Generally, when I talk about membrane proteins, more specifically, I talk about transmembrane proteins, which come in three different types. You have the single alpha helix, you have the multiple alpha helices, and then you have the multiple beta sheets. So generally, these proteins, they insert into the membrane, and in the case of multiple beta sheets, they form a barrel-like shape to form pores in the membrane, which exerts exact control over the fusion of small molecules over the membrane. And generally, these multiple alpha helices, what they do, they perform functions as receptors. So they can interact with other proteins or biomolecules, change conformations, and activate some kind of signaling pathway. Even though only 1/3 of the human proteome are membrane proteins, more than half of the drugs target membrane proteins. So they're very important. And besides that, there's also something funny going on, where with regular proteins, when it's being produced by the ribosome, you have your hydrophobic and your hydrophilic residues. What generally tends to happen is the hydrophobic residues, they tend to cluster together, and this is the main, the driving force behind protein folding is the hydrophobic effect. So you can think of this as having a drop of oil in water. This drop of oil tends to stick together. It doesn't like to interact with water through the hydrophobic effect. So a soluble protein, what we generally see is we see a hydrophobic core and a hydrophilic surface. So it becomes soluble and doesn't aggregate. However, with membrane proteins, we kind of see the opposite, because it's inserted into a hydrophobic membrane. So what we observe here is that we have hydrophobic residues on the surface, which generally interact with the membrane. And in the core of the protein, we have this weird mix of hydrophobic, hydrophilic residues, because all of a sudden it can create this complex hydrogen bonding network in the core, because there's no water around, so it can't be disrupted by water. So now all of a sudden this is the main driving force, or one of the driving forces behind protein folding. And besides that, when we take a look at the SCOP database and we take all the different protein families and we take a look at the soluble proteome versus the insoluble proteome, we find that some protein folds, like the IG-like fold and the TIM barrel, they are unique to the soluble proteome. You will not find them in the membrane. Some proteins, like the beta barrel, you can find them in both the soluble proteome as in the insoluble proteome. So you do find some beta barrels in the membrane. And then there are these unique topologies, such as the GPCR, which are exclusively found in the membrane. And this raises some very interesting fundamental questions. Why don't we see these topologies in the soluble proteome? Is it because the dynamics in the membrane are so different that these proteins, they cannot exist in the soluble proteome? Or is it just that these topologies, they have not been explored by nature, or there has not been an evolutionary pressure or the need to develop these protein topologies in the soluble proteome? So yeah, we are protein designers, so we thought, okay, can we design some of these complex topologies of both the soluble proteome and the insoluble proteome and design them as soluble proteins? And especially when we focus on the insoluble proteins, we can answer the question, okay, maybe we can get these topologies from the insoluble protein to the soluble protein and have all kinds of different useful applications for them. And since this is a talk about AlphaFold, we're going to talk about AlphaFold. So 2 years ago, I was 1 year into my PhD, and then AlphaFold dropped. And the first question that we asked ourselves is how can we use AlphaFold for protein design? So one thing you can do is you can invert the AlphaFold network to do protein design for you. And one way to do that is through error backpropagation, which I'm going to explain now. So generally what you do is you take a random sequence, which is a one-hot encoded matrix. So this is length 20 by the length of the protein, because you have 20 different amino acids times the length of the protein. And this is what you input into AlphaFold, and this perfectly fits with the talk Arne just gave. So this goes into the EVO former, then goes into the structural module, and this predicts a structure. So if we start with a random sequence, we get a random structure. Now what we can do is we can take the structural loss and calculate how far off we are from the structure we want to design for. So in this case, it's the phase loss that AlphaFold uses. So you can think of this as an rotationally invariant RMSD, a more accurate version of it. And then we calculate the derivative of the input over the phase loss, and this gives us an error gradient. And again, this is 20 by the length of the protein, so it tells us which amino acids to change and what to change them into to come closer to our target structure. And we track this error gradient in a PSSM, which is a position-specific scoring matrix, which is again 20 by the length of the protein. And you can think of this as a probability distribution that we're keeping track of. That is, again, for each amino acid or for each position, we keep track of the most likely amino acid that we can find there. So we sample from this PSSM and we iterate this. Basically, this is exactly the same way how you would train a neural network. You would have a loss function, you would calculate your derivative of your input, but then over your weights, and you would update your weights. However, what we do is, so when you train a neural network, you fix your input and you allow your weights to be updated. We do the opposite. We fix our weights and we allow our input to be updated. And in machine learning, this is generally called network dreaming. I don't know if you've seen the images, but if you ask a neural network, like what does a basketball look or what does a human face look like, and you see all these horror images of what a neural network thinks it looks like, it's exactly the same thing. In protein design, we call this hallucination. So we let AlphaFold hallucinate proteins. So first thing we did is, this is also not a new idea. This has been done before, especially with TR Rosetta, so the same method that Arne mentioned in his previous talk, but it hadn't been applied to AlphaFold 2 yet. So we tried to be the first. So we took several different protein topologies, the top 7, the protein A, protein G, ubiquitin, and 4H. And we put them in AlphaFold and we designed sequences for them. And they more or less all looked like the protein I'm showing right here, where we colored in red the hydrophobic residues. So if you remember from the protein folding 101 slide I just showed, generally you would expect hydrophobics in the core, which there are, but here there's a lot of hydrophobics on the surface. So this doesn't make sense at all. So these proteins designed by AlphaFold, none of them worked. Even though they had great PLDDT scores and AlphaFold would think that they were folded. We have several reasons to believe why AlphaFold is such a horrible designer for proteins. One of them is that during training, 10% of the input sequence is masked out by AlphaFold, which makes AlphaFold very robust, but also very a horrible sequence designer, because you can get 10% of your sequence wrong and AlphaFold will still fold your protein, in theory. And the other thing is that AlphaFold has no idea about protein surfaces. Like Arne mentioned in his talk, you only show single chains to AlphaFold. So AlphaFold doesn't know what a protein contact is, a protein-protein surface, what it looks like, or what a regular protein surface looks like. So these are all the things that could go wrong. So we decided to use Rosetta, which is a protein design tool to redesign the surface to become completely hydrophilic. And then some of the designs started working, but still it wasn't great. And in the meantime, a new tool came out, which is called ProteinMPNN. So now our pipeline is updated a little bit. So we still have our AlphaFold backpropagation, which generate a bunch of different variations of structures with sequences. And then we have ProteinMPNN. And what ProteinMPNN does, it takes an input, it takes the backbone as an input, converts it to a graph, and it takes a look at the nitrogen, the C alpha, and the carbon atoms, and then it performs something called iterative decoding. So it first encodes the structure into the network, and then step by step, a random residue is picked, and the network is asked, what is the most likely residue you think should go here? And then it does this again and again, and each iteration it starts assigning residue identities. And this actually worked really well. This was shown by the Baker lab that all these AlphaFold generated designs, they can be rescued through ProteinMPNN. So ProteinMPNN generates a pool of designs. So the way how I see it is we use AlphaFold to generate a bunch of different backbone structures, then MPNN to generate a bunch of different designs, and then we put them through AlphaFold, and we select our best designs based on RMSD, so structural likeliness, structural confidence, so the PLDT in this case, and sequence diversity. We don't want to sample structures that are only 1 point mutation away. We want to really sample a diverse part of the sequence space. But now you can ask me, but Casper, why do you even use AlphaFold? Why don't you use ProteinMPNN from the beginning? That's where I need this slide to explain this. So here on the y-axis, you see your sequence recovery. And on the x-axis, you see different methods. So if we take our crystal structure, we get about 80% sequence recovery in the core, but only 30% sequence recovery on the surface, which kind of makes sense, right? Because the core of the protein is tightly packed, and there's only a certain amount of puzzle pieces that you can put there to finish this puzzle. Which is actually something we definitely do not want with membrane proteins, like I just mentioned, because if it's going to recover these hydrophilic residues in the core, then our protein is not going to fold. Then we use a backrub and an MD simulation. So backrub, it kind of wiggles the backbone atoms a little bit. An MD simulation, it does the same, but more physically realistic, I would say. But we still get very high sequence recovery in the core. But then when we use AlphaFold, it really starts redesigning this core of the protein. So when we just use the AlphaFold backpropagation, we term AF2seq. We get a completely different core, and when we then pass it through MPNN, again, we're going to be about 20% sequence identity. So it's a completely different protein. All right, so now we're ready to redesign some membrane proteins. So in this talk today, because of time constraints, I would only like to focus on the claudin, which is a protein that interacts with other claudins through these beta sheets that stick out, and they form tight junctions. So this is how cells stick together and form a semi-permeable layer of cells. And I would like to focus on the GPCRs, which is one of the most important receptors in the human body. So initially, we were just playing around with taking these membrane proteins and redesigning them. And then something kind of expected, but also unexpected happened, is when we put it through AlphaFold and we redesigned it, it started redesigning it as a soluble topology, which kind of makes sense because AlphaFold is trained on the protein data bank. And the protein data bank, even though a third of the human proteome is membrane proteins, only 2% of the proteins in the PDB are membrane proteins because they're super hard to crystallize or determine their structure. It's become easier now with the arrival of cryo-EM, so I think it's a bit more than 2%, but I need to check. But then when we take this structure of the GPCR and we put it through ProteinMPNN, it all of a sudden it's redesigning it again as a membrane protein. And here, the red dotted line, it is the wild type GPCR that we picked. So it's really making it hydrophobic again. Which makes sense because ProteinMPNN, it was trained on sequence recovery, so it's completely overfitted this model. All it wants to do is recognize what fold it is and just remember what it has seen before. So one trick we did is we took MPNN and we added a bias on the decoding step. So we added a bias towards sampling hydrophilic residues. So you see that there's indeed a shift of the distribution towards a more hydrophilic protein. But still, we weren't impressed. And now it happened that Martin and me, we were at RosettaCon. So Martin Pachessa is the other person on this project, and we spearhead this project together. And we met Justus de Paris, who is the first author of the MPNN paper. And we were having a beer at RosettaCon, and we discussed the idea like, hey, can you maybe retrain this, this model for us, but without any membrane proteins in the training set? So yeah, I think this is one of the great success stories of why we should still have in-person conferences, and also why we should have free beer at conferences, because good things can happen. So yeah, we got soluble ProteinMPNN, here termed ProteinMPNN sol, and what you can see is indeed now we have very much a distribution that is generating very soluble proteins. So we selected our best designs based on confidence, RMSD, and sequence diversity, and we ordered them. So as you can see, only one of the AlphaFold sequences kind of worked. So it was expressing in soluble, but unfortunately, I don't think it was folded. MPNN bias actually did surprisingly well for the rhomboid protease fold and for the GPCR-like fold. But for the Claudin fold, here termed CLF, it wasn't great. But the soluble ProteinMPNN, it got on average a 70% success rate. So this did really well on all of the protein topologies that we tested. So yeah, we tried it. We took some of these most promising designs and we seeded for crystals. And indeed, we got crystals within 3 weeks. I don't know if other people have noticed this, but we have noticed that if we make designs with ProteinMPNN, they crystallize like crazy. So either I'm the luckiest PhD student around, or there is really something about this ProteinMPNN that makes really easily crystallizable proteins, which kind of makes sense, right? Because it's trained on protein crystals. So it might unconsciously or unexpectedly design for good crystal contacts. So here we see the crystal colored versus the design, CLF4 in gray. And we got sub-angstrom accuracy of the C-alpha atoms. So we were really, really spot on, which was super surprising for us. And this becomes also very obvious when we take a look at the full atom RMSD. So as we also take the side chains into account, we only get 1.3 angstrom RMSD, which is crazy. And this can be observed when you look at the inside of the protein. You see it's really, really just spot on the prediction and the crystal. And on the surface, there's more variation, because on the surface, of course, you have more degrees of freedom. So this is definitely harder to predict. So this makes sense. And when we compare it to our initial fold, we're about 3.6 angstrom away. So here in colored, we have our crystal, and in gray, we have our target fold, which is the crystal we used as a template to design this, this structure. And we got the same with the rhomboid protease, but also with the GPCR. So we got a GPCR-like fold, which had sub-angstrom accuracy on the C-alpha atoms and 1.4 angstrom accuracy also with the side chain. So again, what we observed in the core, it was spot on. On the surface, a little bit less so, but still very, very close. And we were about 3.5 angstroms away from the GPCR we used as a target template. So this was what the original membrane protein looked like we used as a template. So if we look at the surface, you see here in brown the hydrophobic surface and in blue the hydrophilic surface. This is what it looked like before, and this is what it looked like afterwards. So we really designed this transmembrane part, which is usually in the membrane. It's completely soluble now. Big disclaimer, this is not a GPCR that we designed. This is completely non-functional. This has more in common with a brick than with a GPCR, because you can boil this protein and it will not unfold. It is, just to make that clear. But now this is, of course, the next step is, can we bring back function to these things, right? Because it would be great if this is a tool for solubilization of membrane proteins. So let's start with baby steps. So the first thing we did is we took the granulin receptor, which has this loop to which we know an antibody binds. So we actually used fold-seq to find something similar to our GPCR-like protein crystal, and we got a hit with the granulin receptor, and we took this epitope from the granulin receptor from which we knew the antibody binds. We put it back on the or we literally graft this, which basically means we transplant the sequence back into the GPCR, and we predict with AlphaFold back whether this protein still looks okay. And we pick our best designs, and we're going to test them in vitro, whether this now starts binding the antibody. And this is indeed what we saw. So we tested several different designs. And all of them, they start binding the antibody when we transplant back the loop. So this is the SPR experiment that we did. So here on the y-axis, you see the response. And generally, when something starts binding, you get a peak in the curve here. And the steeper the curve, the higher the K-on. And the slower it detaches here, this slope here. The less steep it is, the higher the K-off. And yeah, we got about around micromolar affinity for our transplanted back designs. So this could potentially be a tool. So you can think that if you have this GPCR that is soluble and looks very much like a target GPCR, that you transplant back a loop, and you can use this for example, antibody development, which becomes a lot easier than when you have a membrane protein stuck in the membrane, because it's super hard to work with. First of all, you get low expression levels because there's only 2D expression in the membrane, right? Whilst solubly there's 3D expression. Secondly, because of the hydrophobic nature of membrane proteins, they're just really hard to work with. So now let's go a step further. So these GPCRs, they don't just have loops that antibodies can bind to, but they're actually a lot more complex. So G-proteins, they are these 7 helical bundles that are stuck in the membrane, and then they have G-protein which attached to it, or G-protein nearby, which exists out of G-alpha, G-gamma, and G-beta. Now, generally what happens is you have a small molecule that activates the receptor. GDP gets replaced by GTP. And then a signaling pathway is started, because the GPCR, it has a small conformational change that allows G-alpha to start its signaling pathway and G-gamma and G-beta to start its signaling pathway. I don't know if there are any GPCR experts in the audience, or any caffeine experts, because this is a caffeine molecule. In reality, caffeine does not activate the GPCR receptor, but it actually inhibits the GPCR. I only discovered this after I made these slides. So this slide is wrong in terms of the small molecule that I'm using, but the idea is still there. So in reality, it more looks like this. So we have a GPCR in an active conformation and a GPCR in an inactive conformation, where in the active conformation it can bind G-protein, in the inactive conformation it cannot bind G-protein. And generally, their RMSD is between 3 to 5 angstrom between the active and the inactive conformation. So you see here that this helix can kind of open up to allow the G-protein to bind here. In this case, this is not G-protein, but this is something called G-mini. So it's a synthetic construct of G-protein that is just way easier to deal with and way easier to do your experiments with. So this is what we are using. So you have your G protein in its, you can think of this as an equilibrium reaction. You have part of the G protein in the inactive conformation and part of it in the active conformation that can bind G protein. And without the presence of an activating ligand, the equilibrium is going to lie all the way to the left here. So the GPCR is going to be inactive, but in the presence of an activating ligand, it's going to move the equilibrium all the way to the active state. So now baby steps. Can we design, do we have the accuracy to design with AlphaFold the active and inactive state? So can we design a GPCR that can bind G protein designed in its active conformation, and can we design the inactive conformation that does not bind G protein? So we used the following pipeline. We selected a CXC2 receptor, with the main reason is that we have a crystal in the active and inactive conformation, so we know exactly what it looks like. And we fix the motif, so we fix the residues, the amino acids that interact with the G protein. Then we put it through the AlphaFold 2 MPNN pipeline whilst keeping these residues fixed. And hopefully we should end up with a soluble GPCR. It's not a GPCR because it cannot bind any ligand. All it does is it binds G protein. So we will test this in vitro. Now, secondly, can we design for the active conformation, also keeping the same residues fixed, we keep fixed for the inactive conformation, and we put it through AlphaFold MPNN, and then we should get designs that start binding G protein. So we will test this. So what we would expect is that we get an active conformation that can bind G mini and an inactive conformation that actually does not. So we actually did these experiments last week, so I'm very excited to share with you the results. And this is indeed what appears to be happening. So we have our inactive conformation of the GPCR-like fold 13, where we fix the G protein binding residues. And here we see, when we do an SPR experiment, indeed no binding curve. But when we design the active conformation and we test for G mini binding, we get a response. So this is very exciting. Even though the binding isn't great, it's about 36 micromolar affinity. It's unmistakably that there is binding. However, it's hard to compare to the wild type because we don't really know what the wild type affinity is, because generally G proteins are anchored into the membrane. So it's hard to compare. So now we know that AlphaFold is capable of designing the active and inactive conformation. So next thing that I want to be working on is indeed, can we make this protein now switch states? Can we find a sequence that can occupy both states? But that's next. Now finally, I would like to end with the Claudin protein. So claudins, they're inserted into the cell membrane, and they form these huge complexes. They can interact with other claudins, and they form tight junctions, which are semi-permeable, I would call them semi-permeable complexes that only allow a certain type of biomolecule to pass. So this is what they look like. They're 4 helical bundle with 5 beta sheets that interact with one another. And these claudins, they can interact with something called enterotoxin. And enterotoxin is a protein that is being produced by a lot of bacteria like Streptococcus. So you have this bacteria, for example, in your gut, and what Streptococcus likes to do is produce this enterotoxin, and then your tight junctions, they open up, and it can slip through, and if it's lucky, it can steal some of your the resources your body worked so hard to generate. Then what it wants to do is produce more enterotoxin and open up your blood vessels, go into the bloodstream, and produce even more enterotoxin, open up the blood-brain barrier, and enter your brain. So it's generally not a protein you want to have around. But we tried to solubilize these claudins, and we wanted to test if this could maybe be a screening method to check for enterotoxin binding, to see which types of claudins bind enterotoxins. So we took our human claudin 1. We actually tested it on human claudin 1 and human claudin 5, if I remember correctly. We fixed the functional motif again, so these are the residues that interact with enterotoxin, and we redesign the 4 helical bundle part with AlphaFold and MPNN, and we select our best designs again. And finally, we do our SPR experiment, and we do indeed find that that these designs now, they start binding enterotoxin. But not only do they bind enterotoxin when we fit, I know this is a horrible fit, but when we fit our KDs to this, we find that the affinity actually is similar to wild-type affinity claudins. So this was pretty exciting to us. Yeah, these fits look horrible. So one thing we noticed what ProteinMPNN is doing is that it's making very charged surfaces. So somehow we think that the claudins are sticking together for some reason, and that there's something else going on. So we're trying to figure this out. Yeah, we just recently got this data, so this is not ready for publishing at all, but it's unmistakably binding with similar affinity to the wild-type protein. So to conclude, what we have now is a generalizable approach to various membrane protein classes. So this should be applicable to many different protein classes to solubilize them quickly and easily. And more importantly, we can just do this in E. coli. We don't need any eukaryotic cells to work with. So all these proteins, they were expressed in E. coli without any special buffer requirements or refolding of the proteins. With very high accuracy and success rates, E. coli is generally easy to work with. So what can we do next? So the next thing that we would like to do is try to introduce conformational switching. So we have shown that AlphaFold can design for the active conformation in GPCRs and the inactive conformation, but can we now maybe design a sequence that can occupy both states? Which would be really cool to start designing a synthetic GPCR. Next thing is instead of fixing the G protein binding site, we can fix the ligand binding site. And this would be very interesting for the pharmaceutical industry, for example, because what they do is they take these receptors from the membrane and then they screen it against a library of 1,000 different components, and they see what kind of chemicals would bind to this receptor, and this could potentially be used as a new drug. So what would be easier is if you solubilize the receptor, keep the ligand binding site fixed, and then use this for drug discovery. It's easy to get a lot of protein in high concentrations, and generally soluble proteins are easier to work with. We're actually testing this part of the pipeline next week. We made a bunch of designs for ligand binders, so GPCRs that bind both peptides and small molecules. Yeah, so I'm very nervous about next week, so fingers crossed. I hope that that goes well. Then thirdly, now it's time for me to hallucinate a little bit. We can go even further. So one of the very interesting GPCRs is a GPCR called rhodopsin, which can be found in ice. So you have rhodopsin that has a small molecule in there, which is sensitive to light. So when it gets hit with light of a certain wavelength, it changes conformation, which allows the GPCR or the rhodopsin to change conformation, which starts activating a signaling pathway. So can we solubilize these rhodopsins and then have some kind of optic switching effect, which would be really nice and very useful for many applications. And finally, vaccines and mimetics for antibody generation, so we can transplant back extracellular domains of these receptors and then use these for antibody generation. For example, you can use them as vaccines, so antibodies are being created against certain epitopes of this protein, or for example, you can immunize mice with them, and then take the antibodies of mice and have antibodies that target a specific GPCR in the human body as a therapy. So yeah, first of all, I would like to thank my my professor, Bruno Correa, who's helped me get all this research on track. And most of all, Martin Pachessa and Nicolas Goldbach, who are with me on this project and are carrying their weight just as much as I do. And yeah, I couldn't have done it without these people. And as a special thanks to Alex Vecchio, who is an expert on the field of claudins, who helped us with the binding assays of claudins to the enterotoxin. So yeah, thank you very much for having me.